Hello all. I'm going to tell you how we can model Drosophila to study human diseases. And I'll be mostly talking about three strategies with the examples. So it's all about Drosophila. And so Drosophila story started in 1916 with the discovery of white mutant and non-disjunction that proved the chromosomal theory of inheritance of heredity. And then after that, since then it has been used to study many different things like basics of developmental neurobiology, behavior, regeneration. And also now we are using Drosophila to study many different diseases and disease pathologies, which include metabolic syndromes, aging, cancer, and also neurodegeneration. So why Drosophila? So Drosophila genome was sequenced in 2000, and it's about 180 MB, out of which about 120 MB is the eukaryotic portion, which contain protein coding genes, which are about 13,919. And now out of these 13,919 genes, about 60% are conserved in human. And out of these 60%, there are 50% that are actually essential for survival, means loss of these function of these genes lead to lethality in Drosophila, which means they must be essential ideally in human as well. So we'll see that. So and about 75% of all these human disease causing genes are also conserved in Drosophila. So suppose there are like 2,000 genes that cause disease in human. So 75% of them are conserved in Drosophila, which gives us a very good, chance to study their basic mechanism in Drosophila and then see whether they behave same way in Drosophila. So this is one big reason why we choose Drosophila to model different diseases. Another big reason that you heard yesterday is the availability of genetic tools, and also there are like constantly increasing tools for genetic manipulations. So combining these two things, we normally take so there are three approaches that people take for modeling disease in human disease in Drosophila. The first one is reverse genetic approach. Then second is forward genetic approach. And then now a new one that people are using is diagnostic approach. So basically, what is reverse genetic approach? So reverse genetic approach starts with where we already know which gene is causing disease in human. So since we have this information, what do? We go and find out whether there is a homolog of this gene in Drosophila. And once we find a homolog of this gene in Drosophila, then we'll try to perturb its expression. We can make hypomorphs, or we can completely delete the gene, or we can overexpress depending on how it is in human. And then we'll perform complete phenotypic analysis. We'll find out which cellular and molecular processes are being affected in Drosophila, and we'll see whether Drosophila show the similar phenotypes as human symptom. Okay, so how do we do that? So first step is to basically, I'll just give you a brief of all three methods. The second is forward genetic approach, where we do not know which gene is causing the disease in human, and we do not know whether any Drosophila gene can actually cause disease in human. So what we do is people take unbiased screen for identifying new mutations that are linked to specific phenotype. Let's say to find out genes that are linked to neurodegeneration in human. So a large scale screen which is being performed in Drosophila to find out genes that are causing neurodegeneration in flies. Then from those, the screen will be designed, which tomorrow's session will cover the screen, but I'm just giving you an overview. So this screen was designed keeping one phenotype in mind. As I said, neurodegeneration. So let's say screen for all the flies where we see neurodegeneration. All the flies are degenerating, or in all the flies, neurons are dying. So we shortlist the candidates out of, suppose we started with 1,000 genes and we screen, and only about 20 genes show us neurodegeneration phenotype. What next? Then we don't know yet. We just know the lines. So we'll find out which gene is it that is causing neurodegeneration. And once we know which gene is causing neurodegeneration, we'll find out whether this gene is conserved in human or not, and whether it has any link to neurodegeneration in human. And this altogether could be a new gene that is causing neurodegeneration in human, which is not studied yet. Okay. And the third approach is the diagnostic approach, where there is data available from patients where their whole genome, excuse me, Manish. So where the patient's whole genome is sequenced or exome is sequenced, and then we know we find the potential candidates where, I'm sorry. So there were patients whose genome was sequenced or exome was sequenced, and these patients have some rare disorders which we do not know what causing these disorders. So after this whole exome is sequenced, we find some potential candidates. Suppose there are like 10 or 15 candidates which could be different in a patient than the normal sibling. Now, we have these potential candidates. So what do? We cannot look at these candidates in human. We need to find a host. So we go to flies and find out whether these genes have any homolog in Drosophila for all these 10-15 potential genes. Then we will create we'll cause gene disruption, or we'll create somehow perturb the gene expression. At the same time, you also want to find out whether this gene which is there, which is conserved in human and fly, whether there is a functional conservation. The gene sequence is there doesn't mean they are functionally conserved. So we have to check that. And then once we know what is once we know that the function is conserved, then we are going to look at in detail their phenotypic, we'll characterize them phenotypically. We'll find out which protein it is, where it is involved, what is its function molecularly, and in which cellular pathway it is involved. So these are the broad themes that people use to identify new genes that cause disease, or take the information from patients where we already know the disease, but we don't know the candidate, or we know the candidate, we know the disease, but we don't know how these genes or proteins are working in cells. So we'll start with the first one, the reverse genetic approach, which is very commonly used approach, mostly for neurodegenerative diseases like Alzheimer's, Parkinson's, Huntington's, and ALS, where what people did, we already know the information which gene is involved. We already know whether this gene is overexpressed, or it is a loss of function mutant, or it's a totally gain of function, means there is a new mutation which gives totally new function to this protein and abolishes its original function basically. So how do we do this? As I said, that we already know that the genes we have, we know whether they are conserved in Drosophila. So how do we find out what is the homolog? So for that, there are several public databases that you can use to find human homolog of Drosophila. I have listed a couple of these here: Gene to Function, Hugo Gene Nomenclature Committee, Entrust Gene, ClinVar, and Human Gene Mutation Database. But there are many more that one can use. I normally go for the first one, Gene to Function, which was developed by Norbert's group. And it's a very nice site. It's very easy to understand, and you can work with it easily. So how does it look? This is the web page. This is how it looks like. Now I'll just change my pointer. So now if you look at the left side here, it says search by gene and species. So when you want to find out whether the human gene is conserved in Drosophila, so you just put human here and you put down the name of the gene that you want to select. So I selected part 2, which is, I have mentioned here, part 2 is implicated in Parkinson disease, where mutation in Parkinson have shown to cause Parkinson disease. Like people have motor problems, they have neurodegeneration eventually. So let's select Park here. So I selected Park here, and then I'll just say search by gene. What I also can do is, if I don't know which genes are involved, I'll simply go by the second search here, which is on the right side. Search by disease. Okay. So you put down the name of the disease. Let's say Parkinson disease. So I put down the name Parkinson's and I search. Now I searched here and then it gave me a list of database. What are these? So there are many different databases. People have actually screened patients where they actually have potential candidates. Maybe not all of them are linked to Parkinson, but they have potential candidates. And there are different categories like Parkinson disease based on age of onset, Parkinson disease based on like whether it is familial or sporadic, and then Parkinson disease where you have motor problem or a cognition problem. So you see that there is a late onset. So there are many different candidates based on that. They have divided this disease into several. So now I clicked on 2 here, Parkinson disease 16 and Parkinson White syndrome. And then I just said search. So I'm interested in these two because somebody suggested, okay, these are the candidates genes that we want to study. We don't know much about these candidate genes. So I just enter and it gave me Park 16. Now, Park 16, I enter and this is the web page that I see. So we gave just the name of disease or we give the name of gene. It will lead us to this page. Now this page here basically will show you the name, the symbol, and here the species name. So here basically you have all the organism where Parkin gene is conserved. Then, so we see already there is fly here. So not just this, it will also give you other information like what is the specific gene ID for search in fly base. Then what is the score? Like score means here whether this gene is conserved in fly and also in humans, so the score is good. So there are many different ways they have actually looked at this score, and then at the end they give confidence level. The confidence is high here, means yes, it's a true homolog here. So you see here it says low confidence, means it's not that great conservation, but in case of fly it is good conservation. Now you see here you can also find out all the other information about Parkinson because they have listed the publications also. Then are there any known function associated with? Yes, there are 2 at least function which people have seen or have reported. Then what are the cellular processes it is involving? That is also mentioned here. Interacting partner, genetic interactions, and whether there are RNA lines or whether there is a structure available or at least 3D, and then whether there are cDNA clones available and how many people are actively working on this gene right now. So this information, all this information you'll find in this gene to function site. You just enter the gene of interest and you get this information. Now, you can also see the pair-wise alignment, means how good the conservation is. If you want to see in human, suppose there is a amino acid which is at the position 100, that is mutated to, and as a result we have Parkinson disease, whether that a particular amino acid is conserved in Drosophila or not. So you just click on this and you'll find it out. Okay, so here is a fly which we see good conservation, and this is how we look at the alignment looks like. So as I said, it will give you a nice alignment of human protein and Drosophila protein. And then you have, if you scroll down, which I can do here, but if you scroll down, you'll find out what is the meaning of these different 2 dots, single dot. So basically it's the same amino acid, same family, you know, if there is a charge difference, so it'll list down everything. So now what we found so far is like, okay, we know there is a database where we can go and find all the information. And this database is actually giving us the homolog and the score, whether it is a good homolog or not. Okay, so we found the homolog. What is next? Now we want to alter the expression of this gene. Okay, so for that, we'll basically start. So Dipti taught you yesterday how many different methods are there for creating you know, gene mutations or knocking down genes or knocking down proteins and all these things. So I'll just briefly tell you. So once we have the gene, first thing we need to know is whether there are mutants already available. In Drosophila, we have a whole bunch of different lines available. That could be transposable element insertion that is disrupting the gene function, or it could be a point mutation that came from EMS mutagenesis, or you have the transposable artificially made transposable element based gene trap lines. And all of these, I mean, you can use these that are already available. In case you don't find that, a source of mutant, then you can either do RNAi or gene deletion by CRISPR, or you can do protein knockdown using degradFP, or you can do targeted disruption, where you can actually exchange the sequence of this gene with another sequence which contains the point mutations. Also, one thing that is important that people have done in most of the neurodegenerative diseases, that is, you can express human disease variants using US Galfo system. Okay, so I'll tell you about US Galfo system in a second. So suppose for that, what we do to find out whether there are already available mutants are available, we go to flybase, and in flybase we look for the lines. So here I see there are many lines. I see this line here, TRIP, which is a RNAi line, and I see here there is a mutation. I see here there is a protein tag line. So all these lines that are available, they're all listed in flybase. So you can easily pick them or order them and then start working. So as I told you that most of these diseases that I have listed here, neurodegenerative diseases, in majority of these, or if you see all of them, actually have one way where we have to actually express human protein. So in Alzheimer's case, the majority of models, Drosophila models that are developed, they are done using or overexpressing human tau protein or A beta 42 protein, which is overexpressed in Drosophila. Okay. For Parkinson's, of course, the Parkin mutant, but apart from Parkin mutant, this protein, which is called alpha-synuclein, which you don't find in Drosophila, but it's there in human and it is implicated in Parkinson, that you can overexpress in Drosophila, and it actually gives you similar pathological, phenotypes or phenotypes that you see in humans. Same for Huntington. Drosophila has Huntington gene, but the wild type doesn't give you a mutation that it doesn't give you, it needs a specific mutation, which is basically a stretch of polyQ gene that is overexpressed in flies. And that gives you hello, can you hear me?
Yeah, you were muted in between, Sonal.
Okay, so yes, Huntington gene, which actually has just alone, it will not, overexpression of just Huntington doesn't cause any phenotype, but when it is expressed with an extended stretch of polyQ, it gives you phenotypes like Huntington disease. And for ALS, we have VAP-B or overexpression of plus mutant version. So you see here, so not just loss of function gene caused disease, people have overexpressed human mutant version of protein to model these diseases in flies. So that also we can do using Drosophila or US Galfo system, sorry. So how does it work? Basically, the US Galfo system, as I'm sure you have heard in last classes, but quickly, the US stands for upstream activating sequence, and there is a transcription activator. So this system is adapted from yeast, and it works very well in Drosophila. So where Galfo goes and binds to US, and then it will drive the expression of any sequence, coding sequence, which is placed downstream of this US sequence. Now we can control the expression of this downstream coding sequence. It could be RNAi or human gene, using temporally and spatially using gene or tissue specific promoter here. So this tissue or gene specific promoter is giving you spatial and temporal control. of expression of the downstream sequence here. But there is another thing that actually can control the expression level of GAL4, and that is HSP70 basal promoter. So when these lines, the GAL4 lines were being generated, what the people did, they who generated these, they placed HSP70 basal promoter just before the GAL4 sequence. Okay, so HSP70 stands for heat shock protein 70. The promoter responds to heat. So if the temperature is high, you'll have higher level of expression of GAL4, and if the temperature is low, you'll have low levels of GAL4. Okay, so this way we are getting spatial temporal as well as the spatial temporal control of GAL4, as well as we have the different amount of GAL4. Altogether, this will express the expression levels of the human gene that we are expressing or RNA that we want to express. So how do we do? So most of these neurodegenerative diseases that I told you about, they are late onset disease. And these genes like WAP-B and not FARC, but WAP-B and other genes, they're actually also important for development. So what happens if you want to knock down a gene which is essential for survival? You cannot make a mutant, because if you make a mutant, and you might not reach the adult stage. They may die way before. They could be embryonic lethal, they could be larval lethal or pupal lethal. So how to deal with this? So that's where we get this US GAL4 system to work. So the way we do these kind of things is, you collect versions of GAL4 and you collect males of these genes that you want to express. And then you set up this cross, normally in small vials. That's what we do initially. So you set up these crosses and keep these crosses at 18 degrees incubator. And you let the animal develop. So what will happen at 18 degrees? The level of GAL4 will be less, much less. And as a result, the level of this mutated version or RNA will be much less. Okay, so you are not affecting gene. Then you let the animals develop at 18, and when they eclose, you keep them for 2 to 3 days until the development is complete, and then you transfer these flies to 29 degrees. And now what will happen at 29 degrees? You'll have expression of good expression of GAL4 protein. Now you have a lot of GAL4, will go and bind to US and immediately start expressing either the cDNA that you are trying to express or the RNAi. So basically, you're creating a disease situation condition. Now, you express them, you wait for 2 to 3 days, depending on when you start seeing the phenotype. Okay, so first, when you set up this experiment for the first time, you have to wait, do it in a small scale and wait whether you see phenotype, or like which day. You see day 3, you see day 5, you see day 7. So once this is done, you have, okay, it is the day 3 when I start seeing problems in flies. They cannot climb, they cannot walk, they are dying. And then you start your large scale experiment according to this time point. Okay. So you set up this experiment, and now there are several assays that people do for especially neurodegenerative diseases or also muscular dystrophy. So first one is the longevity assay, and then we have locomotion and climbing assay. Locomotion could be like adult fly walking, or it could be larval crawling. Then we have flight assay, whether the flies can fly or not. And also you can look at tissues. So what you do is, either if you have brain or you have muscle, whichever tissue you want to look at, you take that tissue and then look at what is happening. In neurodegeneration, we take adult brain and look at the degeneration. Okay, so I'll just tell you about 2 assays that are mostly done. First is longevity assay. So you are checking lifespan. You are expressing the disease variant. You're overexpressing that, and you want to look, or you're knocking down a gene and you want to look the phenotype. So how do we do that? So as I told you that first you set up the cross. So now for large scale experiment, you need good number of flies, and good number means
So,
yeah.
there are 2 questions. I'll quickly address them. I mean, I want you to address them because this was from
Yeah,
the
I'll see.
last slide.
Okay.
So you can, yeah.
So the HSP basal promoter seem to act like GAL-AT sister. Is it the same? No. So it's not the same. GAL-AT. So GAL-AT is one thing that you think because people use GAL-AT TS. So there is a temperature sensitive version of GAL-AT that is active at lower temperature and not active at higher temperature. Okay, so it sounds like that, but here the HSP basal, just think about it. For GAL-AT, you're adding one more component, and here you are not adding that component. So it works that way, but here you have one less chance gene. So, and GAL-AT you use in cases, so not every GAL4 line has HSP70 basal promoter. The new GAL4 lines that are created, they do not have them. So for that, we need GAL-AT. Otherwise, and GAL-AT also you can use to block certain cell population or certain cell tissue system. So suppose I want to express some, so I'm using actin GAL4. If I'm actin is ubiquitously expressed. So if I shift actin GAL4 at 18, there is not much not much GAL4. I shift it to 29, there is a lot of GAL4. Okay, but the way it works with GAL-AT is now I have actin GAL4 and I have GAL-AT, and now GAL-AT is driven by another promoter, which let's say is expressed in brain. So now actin GAL4 at 18 degrees together with GAL-AT, there is not much expression. Now I shift it to, or at least it is blocked, or there is not much expression. Then I shift these flies to 25. At 25, what is happening? Actin GAL4 is expressed, GAL-AT is also there, it is not inactivated. So actin GAL4 will drive expression everywhere, but the brain specific driver of GAL-AT will try to inhibit its, or it will inhibit its expression in brain. So in a way, you're filtering, you're trying to filter that the brain is not going to get affected because of GAL4. Do you get my point? I don't know, Dinesh? Okay.
Yes,
all right,
I get
so yeah. So yes, the second question is the same, right? It's a GAL4. No, not all the GAL4 lines have HSP70 base. So these are the very old lines that are there. But the traditional ones, actin GAL4, tubulin GAL4, or ELAB GAL4, you know, those all have. The new ones do not have. All right. Yeah. Hello.
Sorry, Sonal. My name is Dr. Razana. I have a question actually. Do you do this HNE staining?
Yeah, I'm going.
Section or whole mount?
Yeah, I'm going
to whole brain mount.
Yeah, I'm going to tell you about that because I have a slide for that.
Are you getting me?
Yes. I'm going to answer your question.
Did you got the question? Hello.
Yes. Can you hear me? Hello.
Hello.
I'll write here.
Hello.
Yeah.
Yes.
I can hear you.
Yes.
I'm going to answer your question. I have a slide about it.
Yes, please.
Yeah? Okay.
No, actually, my concern is that we can have Alzheimer's disease model, we can have Parkinson's one, and other Huntington, etc. But every region, in for example, in rodent we have a specific region for those. Particular
Yes.
For Parkin we have a special region for example, we work on a stroke. We have a particular area in cortex, So yeah. My question is that if you mount whole of the,
okay.
The whole
of
the.
So HNE is, I am saying, just for overall degeneration. But for Parkinson, we also look for DA neurons. Drosophila also have DA neurons, and there are specific markers for that. And you can actually do stainings and look for loss of that those particular neurons.
Okay.
All right. So where was I? Yeah. So how do we do this aging assay? So as I was saying that we have to do it in large scales. Large scale means you start collecting, like you have have like at least 70-80 virgins, and you will take like at least 50 males and put them in a cage. And in cage, these are not vials. We use cages for collection of embryos, like large number of embryos. So you set up this cage and you have to set it up again, initially at 18 degrees. And then you give certain time. So suppose you want to collect eggs for 3 hours, because you want all these flies to come out together. Otherwise, what happens if you keep them, if it is non-synchronous, then you'll have, today you'll get 3 flies, tomorrow you'll get 5 flies, and it's difficult to manage when you are working with large numbers. So just to keep the, have the collection in a synchronized way, you set up a cage, you put all of these flies, and you let them sit there for a day and don't collect for the first day. Then you fix a time. Every day, same time. So I want to collect from 10 o'clock morning to 12 o'clock or 10 o'clock to 1 o'clock. So I'll change a plate in the morning, 9 o'clock. So just to give them a fresh, like some fresh food, so that if they're storing some eggs, then they will lay. And then I start my proper collection at 10 o'clock. So 10 to 1 o'clock, I'm collecting eggs. And now I change the plate again. And the plate that I collected 10 to 1, I'll keep that plate at 18 degrees for a couple of days. So there are 2 ways. Either you take the embryos out from here and shift them to vial, or the way I do it is I keep them at 18 degrees, and then when they start hatching, I transfer larvae into vials. So that way I know that how many larvae I'm shifting to a bottle. So again, at the same time, everything should be same. Every day you are going to go and do the same thing at the same time. You cannot change the time. So 1 o'clock next day. So day 1 I collected. Day 2 I am picking young larvae and transferring them to vials. And now I transfer only 50 larvae to a small vial. So day 1, day 2, day 3, day 4. That way I'll keep collecting. And the day 9 is the day when the first time I get my flies. They'll start hatching. So day 1, suppose only 3 emerge. So I'm going to discard those. What I do is I'll just start collecting adults from day 2. So from day 2, you get good numbers. So suppose I got like 10 flies on day 2, and then day 3 again 10 flies. So that way I'll collect for 4 days when I have good number, like 10 flies every day I have for 4 days. This is my good number. Okay. So this way, for these kind of experiments, you'll have to actually collect at least, you'll have to do, let's say 8 or 9 biologicals, and every time you need 40 to 50 flies to make sure what you see is correct. So now I have these flies. How do I basically keep or maintain this experiment? So for that, I need to transfer. When you collect these flies, keep them with males for 1 or 2 days. Okay. Don't let them sit alone for all the time, because then over time somehow the vials get fungus and then they start dying early. So the way it is done, you collect these 10, 20, 10 males, 10 females you collect, and then you sort them out. So what I'm trying to say is, once you have your synchronized egg collection, then once you have these flies coming out, keep them with males for a day or 2, and then separate them and then keep them in fresh vials. Every other day you give them fresh vials, so that there are larvae, but they are not crowding so much so that females get stuck and die, or males get stuck and die. So these flies, every 2 days you have to transfer to fresh vials. And this way you have to maintain your diary and see until the last fly dies. You have to keep account that, okay, day 15, day 16, and now it's like day 45. In controls, the flies are okay, and day 46, they die. While in mutant, what you see, or overexpression of one of these human gene, disease variant, or the loss of function like RNAi expression, suppose they die on day 20. So you have to keep a record. Even if the controls do not die, you have to go till the end to note it that control died here and these overexpression flies die here. And this is how you can get your data, and you can plot and see how does it look? Okay, so in majority of cases, when people have overexpressed these disease variants or knockdown genes, they actually lead to short lifespan. All right. So this is one way of checking the effect of these. And now you know that they die at age 25, means you have a point. So all your experiments that you want to plan to look at these flies, you want to dissect the brain, you want to look at the plaque, you want to, you know, stain them for particular neurons. You perform all these things in the mid of the time, not too early and not too late. Too late because it will be not a good way, or the information will be completely lost, and initially you may not have that enough information. So a midpoint, like suppose they're dying at day 25, so I'll go around day 13, day 14, and then I will look at, you know, all the cellular processes, or look at whether neurons are all okay, or, you know, whether these flies are walking properly. So all these experiments, I'll set up this lifespan thing first, and then I pick this point, and then the rest of my assays will go according to this lifespan. All right. So the next assay that normally we do it is climbing assay. And similarly, I'll tell you flight assay, the same, using the same slide. So climbing assay. Climbing assay is a very easy assay. So we just need a good glass cylinder, not one glass cylinder. It depends on how many genotypes you want to look at. So suppose you have one control and two experiments, or you have two controls and one experiment. So you have three sets. Then what you do basically is you take three identical glass cylinder that are marked, okay, and you decide a point, which is here it says this is a 20 cm cylinder, which is 250 ml. So you can set it up 2/3. You select a height which is 2/3 of the cylinder. So the normal fly should climb till that point. Okay. Now you put around 10 to 15 flies in that, different genotypes. You don't have to do all of them at the same time. You can do one by one. But just remember, there is a place and a point. All the flies should be in this, like the experiment should be done in the same room, at the same temperature, same humidity, and you're growing these flies at the same temperature with light dark circle. So even little change can give you huge behavioral difference. Okay, so you take the cylinder here, and then you put your flies, transfer your flies with the help of funnel, and then you put paraffin on here. Then you tap it 4-5 times, so all the flies are down, and you have your phone or your camera ready, because it's very difficult when you're watching it like this. You can't count. So you have your recording device ready. Just tap couple of times, so you see they are all there down, so that is time 0. And then you see that they start to crawl up, and then after, let's say, a minute or 2 minutes, you will see they're all are here. These are all control. Even in control, there are some flies that fall and then start crawling again. So you give them enough time. Now, same thing you have to do with your experiment, where you will put them with the help of funnel. You will cover it, then tap them down, and then you see how are they crawling, and you are recording it. So, I forgot to mention the last slide. Here I'm explaining this, but there are nice videos about these assays which I actually followed, and it's really good. So now, once you have your videos there, you'll have to sit. So there are devices where actually people can track their path, which I don't have, but one can buy those things, and you can point, okay, this is a fly, I want to track its path, how far it went. So you can see that, oh, it went till here, and then it roamed around and then fell down. So at least you know it went up. In control and in mutant cases, what you see is they just crawl here, or they just try to climb and they fall down again, and they try to climb and they fall down again. So this is the difference you need to see, and then you need to everything note down, and then look how many flies could cross this threshold in how much time. So time delay is one thing, whether they can go and stay there, that is another thing. Okay, so everything counts here. So this is a climbing assay. Now, using the same setup, I'll tell you the flight assay. The flight assay also people perform And again, the time point you selected with the lifespan, or you start seeing the phenotype, then you wait for 2 days. So you don't have to go to the end, or you don't have to do it very early. So you select the time. And then for flight assay, what we do is we take a little bit bigger cylinder. Okay, so let's say 1 liter cylinder, and then this is the point. Here you put a paraffin and make a small hole, or you can have a funnel, and then you just drop a single fly, and then the fly will go down. But normally in wild type they just go down a little bit, and then they just go and sit around here or here. While in the mutant case, what happens is they either go down here, they try to come up, but most majority of cases you'll see that they come straight down. So one example where I use this method is brook willow. Brook willow is a protein which is involved in synaptic transmission. The name brook willow came because of this, a crash landing, because these flies, when I knock down this gene or protein, the flies cannot fly. They just straight away come down, and that's it. They can't walk. They don't try to climb. They just sit around. So that's another way you can see whether these flies have any problem in climbing or walking. Okay. So we have seen the lifespan thing, climbing assay, and the flight assay. For locomotion also, you do these kind of things. You have an open cylinder from both sides. You put a fly from one side and you close it from one side and let the other side open. So normally wild type flies, they crawl quickly and then they come out. While the mutant, they just stay at one place. So just imagine this cylinder is kept horizontally, where first this portion is locked, you put some fly and then you close it and you open this side, and then normal fly will crawl out, while mutants will not crawl out. And again, you have your recording device and you have to note down all these things, because every single thing is important here. And as I said, the sample size, you have to at least do 10-15 biologicals. I prefer 15 biologicals if I'm taking only 15 flies in each round. And then, so at least you should have like 150 to 200 flies to make sure that what you see is a real phenotype. And again, grow them all at one place, same temperature, same dark cycle, light cycle, and same humidity. And the experiment should also be done at the same temperature and same places. So these experiments after 18, you're doing at 29. So you try to do this, you can't have the room temperature 29, but go for 25. So for all of these. All right. So these are all behavioral assays that we see, that, walking, crawling, for larva, for adult. What you can also do, that I told you, for a quick screen or quickly checking, you can do a H&E staining on brain sections. So overexpression of these genes, in majority of cases, you see eventually neurodegeneration late stages. So again, these are late onset. So what we do, we let the adults grow at 18 degrees and then transfer them to 29 degrees, depending on 2 to 9 days, depending on how the lifespan goes. And then we dissect adult brain, and then basically, so dissect the whole brain, cut the whole brain, not dissect adult brain, cut the whole brain, and then process for paraffin mounting. And then once you have the paraffin mount, you can get them sectioned. And then once you have the section, you go for H&E staining. These are very simple stainings. Normally, we don't do paraffin embedding in lab. We usually send these to some specific course because the reagents are very toxic. So you cannot just start doing it in lab. So there are places where actually they perform this. So you prepare the sample, which is cut the head and then put them in ethanol 70%, and then give it to the person who will actually embed it, and they will embed and they will cut them and put them on slides. And then once you have the slide, you can do the H&E staining on sections. So this is an example that I had done. So here is the control day 3 and control day 10. And if I look at this gene, which actually loss of this gene looks to neurodegeneration, you see already at day 3, I see small bubbles. And on day 10, actually the whole eye lobes and these were gone. So these are the broad information that you can get, that, okay, yeah, this loss of this gene would lead to neurodegeneration. This is the end point. You cannot get much more information from here. So again, this is like day 10 where the flies are almost, not crawling and sitting and not doing much. If I want to perform the real experiments and I want to see what is happening at the cellular or molecular level, I would rather go at day 3, because at day 3 already I see a phenotype type, or maybe on day 2. Okay. So far we just discussed that what are the how you get this information once you know which gene is causing the disease and which mutation is causing the disease, and whether they were overexpressed or the genes were mutated and there is a loss of function, and how to express them in flies to model them for neurodegeneration, and how to perform these assays. Okay, so similarly, people have, so this was all neurodegeneration. So people have modeled Drosophila for muscle degeneration also, or muscular dystrophies. I have listed down some of these here. The majority of these are muscular dystrophy, which was linked to these two proteins, dystoglycan and dystrophin. These two proteins are very important for actually linking for many different processes, and like regulating cell polarity and maintaining actin connection, and also like several other things which I'm not going to discuss in details, cellular phenotypes. But what do we know? We know that mutation in these two genes actually lead to this muscular dystrophy. And there are other things, that there is one where toxic RNA gain of function means certain RNAs that are overexpressed somehow in muscles, and that lead to myotonic dystrophies 1 and 2. There is cardiomyopathy, where again a disease, a deletion mutant is linked to myopathy. And then we have limb-girdle muscular dystrophy type 2K, where mutation in POMT1 gene causes this dystrophy in human. So luckily we have in Drosophila the one homolog of this gene, and that we can model. So we'll go through the same way, that you have these genes, you have this information, you go and find whether there is homolog in Drosophila. Once you have the homolog, again go check whether there are any available mutants or RNA lines, or if you want to overexpress, you see which Galford line you will use. So since this is muscle, so here you are going to use muscle specific RNA, Galford line, which mostly we use either MEP or we use MHC Galford. And both of these have HSP70 basal promoter. Okay, it means we can again plan this in adults. So if you do this during larval stage, they also die. But if you want to see real thing like a human, you want to these are all most of these are like again late onset. So then what you do is the same way, you don't express, you keep the animals at 18 degrees and they shift to 29 degrees after 3-4 days of eclosion, and then you see whether these flies show any problem in crawling, they have climbing defects or flight defects. Okay, so now once you have this model established, then you can go and look for the cellular processes and molecular function depending on where this gene is expressed. So I'm sure that Deepti showed you yesterday how you can tag a gene with a tag like a GFP and RFP. So that way you go forward and you look at these genes, whether they are expressed in muscle, whether they are expressed early in development or late in development, and depending on that you decide when to look for a phenotype for a particular cellular process. Okay, so we covered the muscular neurodegeneration and how do you model muscular dystrophy in flies. So these are all known genes, reverse genetic approach. How do we go for the forward genetic approach, and why should we go for the forward genetic approach? So we know that there are many genes that are already linked to diseases, but there are many cases where we actually do not know which gene is causing the disease, you know. And for that, we can actually take this either forward genetic approach, or we can go for the diagnostic approach. So what is forward genetic approach? So in forward genetic approach, what we do is we pick the collections that screens that people have done already, or we can plan our own screen. Means we'll plan a large scale screen to identify new mutant which is linked to a specific phenotype. So let's go back to human neurodegeneration, or let's think about cancer. Okay, so I want to plan a screen to identify new genes that are implicated in cancer. So how I do that? People can do EMS screen, or you can bring the RNAi collection, or you can take the transposable element collection, and you screen for a cancer phenotype, which is overgrowth. So I'll show you in the next slide how to do that. So let's say you did the screen and you got some candidates. Now you have the phenotypic analysis, you know the candidate genes. These are the lines. We don't know the genes. So then you map the gene. Okay, these are EMS mutagenesis screen, and you got, like, you know, you started with your own name, like, okay, I am SNJ, so I got SNJ1 mutant to SNJ 10,000 mutant, and 565 is the best line which is giving me phenotype, but I have to find out which gene is it, right? So I'll map it. How do we map it? For that, first thing, we can do sequencing to find out where it is nowadays, but initially it was not possible. So right now it's good that you can simply take the sample and go for whole genome sequencing, and you find out where the mutation is. But again, the whole of genome sequencing will give you 10-15 potential point mutation. How do you know which is it? So in that case, what you do, we have in Drosophila large deficiency or deletion mutants. So suppose you got these 10 point mutations and you don't know which one it is. So you know where these 10 mutations are, in which area. So you get deficiency lines that are basically covering these areas. Okay, so suppose there is a gene A and the point mutation is in gene A, but I don't know whether it is gene A or B. So in Drosophila stock center, we have collections where there is a line where gene A and B both are deleted. There is a line where gene X and A are deleted, and there is a line where only B is deleted. So I'll bring these three lines and I will perform complementation. Means I'll take the stock that I made, this line 565, I will cross it with all three deletion, and I see what survives. So what survive means, if the mutation in is in gene A, I cross it with deficiency where A is deleted, this line will die. But if I cross it with a line where only B is deleted, this line will survive. So this way you perform complementation, and then you figure out, okay, my mutation is in gene A. So I'll keep it. So I know now where my mutation is. I know the sequence, I know the which gene it is. So now I have my gene. What I have to do? Go back to the same thing. Find a human homolog. And this time what I'm doing is I'm finding a homolog using the same gene to function. And then I'm going to study that. Okay, so this is usually done using EMS mutagenesis, what I explained, but again, tomorrow we'll have a large screen talk where you will understand it a bit better. So now we did EMS mutagenesis. What if I have a large collection of RNAi lines and I want to find out which genes are involved in brain development? Okay, and in human there are many cases where we see microcephaly, means small brain phenotype, and the kids or babies don't survive, or they die while they are still in, their fetal state basically. How do I find out which are the genes that are involved in brain development? What I do, I have a large RNAi collection, so which is US RNAi. What I will do is I'll select a specific GAL4 which is expressed in developing brain. So since I want to find out which are involved in brain development during development, so I will not keep it at 18. I will from the start keep them at 29 degrees so that I have more GAL4 expression. So again, inscrutable GAL4, which is expressed in developing brain, has HSP70 promoter. Okay, so I'll set up this cross. And this I will set up a small scale. I don't want to do it at large scale for the first time. So I'm collecting 10 virgins. I'm setting up a cross with 5 males. I set it up virgins of GAL4, males of UAS, and I kept them at 29 degrees, and I let them die there. I'll see, okay, this line number 6 leads to lethality. So just remember, when you set up this cross, you set up on 29, you keep it for 3 to 4 days for enough larvae, and then you transfer the parents to a new vial because you want to keep it rolling. So vial number 1, I let them sit at 29, and I just want to let them die to see whether loss of this gene leads to lethality or not. So now I see, okay, line number 6, it is lethal. So I still have the vial number 2 with me. Now I'll take the vial number 2 and I'll take the larvae, and then I will dissect their brain and see what happens. So if I see, oh, I forgot to put the normal brain size. So there is a normal brain. In control, I'll see normal brain. But if these larvae are dying, there can be two possibilities. Either the brain is not developing, it is very small, or the brain is huge. It's just huge and there is no, so the reason could be that the stem cells that are actually giving rise to brain cells, they're just keeping on dividing. Here, they're not at all dividing. Okay, in wild type, they divide for some time, they make neurons, and then they stop dividing. So these are the two conditions we see. Both are both the conditions are important because both are giving us some information that this particular gene is important for development of brain, because this also leads to lethality, this also leads to lethality. Okay, and this way we get good candidates, one which is giving overgrowth and one which is giving undergrowth. Okay, similar way. So here I just told you about inscrutable GAL4, which is expressed in brain. What you can do is you can do some other screens depending on the GAL4 line. Okay, so now just a second. Depending on the GAL4 lines. So like here, if I take a GAL4 line which is expressed in hematopoietic stem cells, and what I want to find out is, I want to find out which gene is important for development of proper hematopoietic system, okay, or blood cells. There is a program for every cell that they should divide for certain number of times for certain time point. If I lose this gene, what happens? So it could, depending on the GAL4, I will screen for phenotype like this. So here, what is the phenotype? This is normal third instar larva, and you see these black dots. These are melanotic tumors. These are caused by overproliferation of a certain type of a certain type of cells, overproliferation of certain type of cells. On the right hand side, what you see is basically, hello? Hello?
We can hear you, Sonal. Sonal, now you are muted. Sonal, you're muted.
Hello?
Yeah.
Hello? Yeah,
Yes.
sorry, this system got hanged, so I couldn't. Yeah. So depending on the GAL4 you use, you can plan a study design. So here I wanted to look at this phenotype. So again, at small scale, first I actually set up experiment in a small scale where I'll take 10 to 15 virgins and 6 to 7 males. And then I'll set up this and I will screen these larvae because now I know what I'm screening for. I'm screening for appearance of these tumors. So, okay, I see it. But this will not give me much information. What I want to do is, I have the gene here. I want to find out what it is doing in this particular cells. So that I have to find out. Similarly, if I want to find out genes that are involved in cancer, new genes I want to search. So I can take a specific GAL4, like I prefer to use wing disk for that kind of screen. So I'll take a wing disk specific GAL4 driver and knock down these genes. So here you see the wild type wing disk, and here you see the overgrowth. Similar thing, similar gene could where we saw in brain overgrowth could be involved in this phenotype as well. So all these things at small scale we first perform, and we get just to get the candidate gene. Now, once we have the candidate gene, we have to go back to the candidate gene and look at this carefully using a specific, like using the specific markers. So we are not going to go into details of cellular and molecular findings, how do we find out, but at large scale, this is what we find. Now, once we know these candidate genes, so we found 3-4 candidate genes from each screen, we'll just go back to the same path, where now we have the candidate genes. We want to find out this fly gene, whether it is conserved in human or not. So in this case, I'm selecting fly as a species, and then I'm putting the gene name, which is an uncharacterized gene here. So the gene is there, whether it is conserved in human or not. So yes, I find this gene is conserved in human. This is the first one. The confidence is good. We know there are like some people are working on this gene in human. We see that there is no molecular function or cellular process is known, or molecular function is also not known. So it's a good candidate to work on basically. Okay, so let's just go and look at this gene here. And once we know that this gene is conserved, we are going to do one important experiment. So what we found out, okay, there are candidate genes that are involved in particular process in Drosophila. Its loss leads to particular phenotype in Drosophila, and it is conserved in human. But whether the function is conserved, this is one important thing that we have to find out for every new candidate that we find in fly or human. So how do we check that? So I assume that you have learned yesterday how to make fly stocks. So basically we create some fly stocks. It's a complicated stuff here. So we want to find out one thing. This is my gene of interest. Loss of this gene leads to a phenotype. So loss of these means both copy loss leads to a phenotype. So my aim is to bring this copy with this copy. But I found out, oh, I cannot do this because they die at embryonic stage. So in this case, I know the lethal phase is embryonic stage. I want to find out whether human protein can rescue this lethality stage. And this is the way I do it. I create one stop where I have my gene with a particular mutation, and I bring a transgene that will express cDNA of human protein. Okay, so this is going to express human wild type protein in this fly. Now I will make another fly where I have again my gene with mutation, and I'm bringing actin gal4 because I don't know where this gene is expressed. I don't know which specific tissue, which specific cell. So I'm going to take a UTS gal4 driver. Okay, so I'm taking here actin gal4. Now, making this fly takes good time. Making this fly also take good time because this is a couple of generation process. But once they are ready, I will expand them in good number because this should be a good number cross. I'll start, so always take the female of gal4. So I'm taking here, let's say about 20 virgins, and I'm taking about 10 to 12 males and a mid-size vial. And then you set up this cross. And what you want to have is, you want to have a condition where you have this mutation in homozygous condition. So this mutation in homozygous condition, you want a copy of this and you want a copy of this. So what happens? This gene mutation over mutation is lethal at embryonic stage, but here we have actin gal4. So the gal4 will be made, it'll go and bind to US sequence, and then we'll start expressing this coding sequence. So we'll express human protein, which is there from the start. Now, if the human protein performs the same function as this fly protein, then these flies will not die. So it will basically rescue the lethality which is associated with this gene. Okay. So we are going to screen for flies that are non-curly, because this is curly, this is, so we are going to screen for non-curly and non-tubby flies here. That gives us that gives us the confidence that, okay, this human protein is working in the same way, it is expressed in the same tissue and working in the same way at cellular and molecular level as a fly protein, and that's why it is rescuing the phenotype. So now our candidate is successful. So we know that we have found a new candidate which is conserved in human, which is causing a phenotype in fly, and then you can go and look for the biology of this, and then you can also do one thing. Now you have the human candidate name, you go and search the disease database. There are many disease databases, or you can contact clinicians and figure out whether they have come across any mutation in these genes that actually causes any disease in human. Okay, so that way you are finding a new gene which is involved in human disease. At the same time, you're going to look at the phenotypic, you will characterize it phenotypically to find out which process it is involved in. Okay, and this goes for any gene, like for wing disc development, cancer, as I told you, hematopoietic stem cell system or brain system, anything you can do a screen like that. So this was RNAi, but you can again go for EMS mutagenesis screen, and or people have done so many screens, and you can pick those candidates and do your own, plan your own screen. So like I planned a screen in my lab for finding new genes using this RNAi knockdown or expression-based screen to find out basically which genes, the new genes that are involved in brain development. Okay, so this can be a large scale or this can be a small scale. You can get 50 RNAi lines and do a quick screen in your lab and get new candidates. All right. So this was the second approach that we can, or people can take to find out new genes that are linked to human diseases, and apart from the already known genes. So you're modeling basically flies for new diseases. Okay, so the third approach, which is very common now, and which is called, which some people call diagnostic approach, which is there to find out new genes that are involved in rare diseases. Like what rare diseases? So there are many people who have some rare diseases. We don't know about those diseases. They could be, genetically inherited, or they could be, I mean, there could be some sporadic cases, but sporadic is little difficult. Most of these are the genetic ones. So what is this diagnostic approach? So people have, hello, there are two questions I see. Are those cryo sections? Not cryo sections. Okay. So this diagnostic approach. So again, there are centers where actually clinicians are working. They get these rare disease, people with rare diseases, they get their whole genome sequence. So I know that at least in IGIB, there are people who are doing this thing. And even here in CCMB, a couple of people are involved in this kind of work. So they have tie-up with the clinicians. So clinicians see patients, they come across some diseases, they don't know how to solve these problems. So they then talk to scientists, what could it be, what could be the reason. So once we have we get their whole genome sequence or their exome sequence basically, then again we come up with, we get certain candidates. As I said, it cannot be single candidate. There are so many different mutations, or the other SNPs we don't know which is actually real mutation or which is actually a just polymorphism. So suppose, as I told you, like let's say we have a case, there are 50 mutants, or let's say there are 5 mutations in this person's, this candidate. So we have these 5 potential candidates. We don't know which candidate or which gene is causing the disease. So what do we do? So in this case, we're not going for a single candidate, right? We are going to look for homologs of all these 5 candidates. And once we know, okay, these are the 5 genes, A, B, C, D, E, and then we'll look for basically whether there are mutants available. If there are mutants available, we'll go and look at a quick phenotype, loss of function phenotype. If we don't have mutants, we'll go for RNAi knockdown, or we'll use CRISPR to delete the gene to find out what is the phenotype of like strong loss of function. And once we have the loss of function phenotype, we'll see whether the human had similar thing. So in this case, let's say there is a kid, he has problem in eye development and his eyes are not developed properly. Now we have these 5 candidate genes, we'll make mutants in Drosophila, and we'll see which of this leads to defects in eye development in flies. While we are doing this, we have generated mutants, and first thing is loss of function. Maybe the loss of function is not causing eye defect, it is causing embryonic lethality in flies, because normally what happens in fly, there is one gene which actually could have several paralogs in human. So in this fly case, we have it leads to lethality, while in human it causes some disease. Okay, so in that, what do? So we did create a loss of function mutant, but we don't know whether we have the same problem or not. In that case, we again go for functional conservation. As soon as we know that human protein rescues that lethality, we can think of other experiments. Then we can again go for conditional knockdown in eyes and look whether loss of this gene in eye development is leading to a phenotype or not. And again, once we know, we are going to characterize it fully and find out whether human variants also cause this problem in Drosophila. So how we do that? So let's go back to the same thing again. We have our gene to function. We found a gene, we look whether it is conserved. Yes, it is conserved in flies, and we pick it and then we look for mutants. If we don't have mutant, we make mutants, and once we have mutants, we go for again the same thing, checking functional conservation. Now this time what we are doing, we are not only checking there is functional conservation by using the human wild type protein. What we are also doing is, we have the information of that potential mutation which we think is causing the disease. So we will also try to rescue this phenotype with a disease variant with that particular mutation. And what happens normally, if this mutation in this gene is causing the phenotype, then normally when we see rescue with wild type, rescue of lethality with the wild type human gene, we will not see the rescue with a disease variant. Okay, so I'll just give you one example of such kind of study that was done in my postdoc lab, and then you will understand how how one person can do this diagnostic approach now to find out whether which gene is involved in disease. So this is a case study from my postdoc lab. I was not involved, but it's a very good study. So what happened is, in my old lab, like they had done screens and they had many different candidates, or they had generated a large collection of mutations. And while they were doing this, there was another center in the same institute that had that is like genome center basically, where they used to sequence people with rare diseases. And there was a couple, a family from Mexico, and they came with their kid, and the kid had microcephaly. So the first kid had microcephaly, he had died, and then they had second kid who also had microcephaly, and they showed really strong like ear problems. So this kid, who looks like maybe 2 years old, but it's actually a 5 to 6 year old kid who was actually not developed. You can see the brain, which is very small. So microcephaly is a condition where the brain is very small. You can see it here. The brain is very, very small compared to normal persons. It's really small. So now this kid has microcephaly, they got his genome sequenced, and then they got 5 potential candidates. So from this 5 potential candidates, in our lab, they found there were 3 or 4 genes. We had mutants for 3 or 4, I think it was 4 genes. We had mutants for 4 different genes, that 4 different potential candidates. So simple thing was, since we already had mutants available, just to look at the brain of developing larvae. So we got these mutants, and then people in my lab, they checked the developing brain. So here you see the control brain, which is the size, and then they looked at brain of many different these 4 genes, and they found in one gene, which is called ankle 2, the brain was small. So this is mutant brain, the ankle 2 mutant brain, which is small. So then they focused on that. Okay, this could be a potential gene. So it is possible that this is the gene which is mutated, which is causing the microcephaly in this kid. So here you have this. When you basically rescue, this rescue I mean is human protein. So when you express human protein in this mutant background, and they found that it completely rescues the size. Not just size, these animals were alive, and then the slice came out and healthy. What they did then, since we knew information about the mutation in the patient or the kid, they expressed mutant variant to rescue this phenotype, and the mutant variant could not rescue the phenotype, which is a small brain in flies. So this confirmed that the kid has a mutation in this gene, and that is why he has this problem microcephaly. What is the next step? So here we got the information. Of course, the next step is you go and find out what this gene is doing, where it is involved, what molecular function or cellular processes. But that is one thing, to understand the function of this gene. The more important thing was then this information was again passed to clinicians. This case came from clinicians to scientists. They looked at this, it went back to clinicians, where they actually looked at they did IVF for this couple, and then selected fertilized egg that did not have, or let's say embryos that did not have this mutation, and now they have a healthy kid. Okay, so overall this shows that Drosophila plays a very important role in actually all these processes, to understand the basic biology and also to find out these mutations or disease causing genes, and to understand basic biology as well. All right, so this was actually the last slide. With this I'll finish. I hope you got some information, and if you have any question you can ask me. Can you hear me?
Questions, people?
Okay, I don't think there is any question. So that question earlier, it was at cryo section. Those are not cryo section. Those are paraffin sections for H&E staining.
Okay, so if there are no questions, I'll ask one question, Sonal. In this particular case, where you are basically looking for these mutations from patients, and they have rare diseases, so it's not, I mean, because there are all these different SNPs that might be present. Every gene, there might be many SNPs. So how, and they may or may not be important. So how people get to these kind of just 4 or 5 genes in the end?
Okay, so I'm sorry, but this is the thing that I think Manish is going to cover tomorrow in detail. So how do you get this large scale thing, and then how can you come to the point and make sure, because that's actually his data, his paper. So he's going to tell you in details about how you come down to bioinformatics. You use bioinformatics and come down to these few point mutations.
I see.
Okay, I'm sorry.
Okay, no.
Uh-huh.
Falak, you want to ask something now? I mean, people can actually
I see there are two questions.
talk and ask questions instead of writing. That will be easier, actually.
Hello?
Maybe I'll ask a question while we wait.
Yeah.
Yeah.
He has a question. He has a question. Seem to have written a question down.
Sorry, I have a question. If you look at the controls in this ankle to mutant rescue experiment,
Yeah.
the morphology of the brain itself is slightly different from that of the controls. I mean,
Okay.
at a cellular level, I mean, even at a molecular level, there might be some changes to that particular, although there is a rescue to it, right?
Uh-huh. So thing is,
And to
if you
another question in continuation to that, so how long do these flies survive? What is the length of their survivability, even if they are rescued?
Uh-huh. So I won't go for this brain image because it's a maximum projection, and it's not that what you see is actually true. That okay, you can't judge from this brain that the cellular level they are different. They are actually same. So they have same number of divisions. They divide. They have polarity, right polarity. They and the flies also, they survive as long as the control survive. They are happy, healthy, and happily growing flies. You could actually establish a stock and keep it. Okay, if there was any problem, then you could see some phenotypes. Yeah. So here, this is just one representative image, which we cannot say that the the morphology looks different. It depends on how we mount. So here we were just focusing on the size, rescue of the size. And this is one image, but they are actually good flies. They are happy, healthy, and they survive good number, like for good time, like controls. And if you look at the identity and similarity of this protein, which is also very good.
Dinesh, you can ask the question while Falak is writing.
Okay. So it was a very interesting talk. So it's very clear that that you can link gene to neurodegenerative diseases very nicely using Drosophila. I was wondering how we could take this to the next level, which is you have a gene which is somehow, let's say here, linked to microcephalization, and from there you would want to find out how is the emergent prop, how is it becoming small, what is, so we have this link between gene and signaling systems, that's fine, but somehow that's affecting the whole brain in a very macroscopic level, which is giving rise to these sort of phenomena. Like you had another image which was really, really good, where you had this brain with holes developing. I think that was Spark. Now I was wondering if Drosophila by itself has some interesting tools to start getting into that. This is the gene to neurodegenerative link. From there, you would ideally want to get to how is that gene actually affecting and giving rise to macroscopic changes in the brain, and how would you try and counter that? So I just wanted to hear a little bit on
that. You are muted, Sonal, if you're talking.
Now?
Yeah, now you're fine.
Somehow it's not moving. Okay, so this gene, SNF4A gamma. So it's a basically subunit of the 3 subunit complex M kinase. So let me tell you how it is, and then we'll see what. So what happens? This subunit senses ATP to ADP ratio, and then when there is no ATP, okay, these neurons do not know. So you are utilizing everything and the neurons do not know. They're just working. There is a point that you don't have energy, and that is when these neurons start to die. This is how what I'm showing here. Okay. So now so the question what I understand is, now we have this end point. How do we go back to his function point? Is that what you were asking? Hello?
Yeah, yes. I think
Okay.
that's sort of, yeah.
Yeah. So see, what is happening? So you know that this gene is very important for sensing this. I knew this information, but nobody has shown that, okay, it could lead to such drastic phenotype. Like in 3 days, you have such bad degeneration. All right. So what I will do now is, what is important? Important to find out whether this gene is expressed in brain, okay, whether it is doing other function, because energy depletion or ATP depletion in 3 days, I am not sure that can give to such drastic phenotype. Maybe it is doing some other thing, and that's for that I have to go back, look at the expression, look at its interacting partners to figure out what is happening, and that will be a huge thing. This itself is a project to find out where it is localized, what it is interacting with, whether it is important in like not just energy sensing. Maybe it's important in some other things also, maybe in polarity, maybe in cell division, because neurons they do not divide. Suppose they start dividing, so especially this complex also has link to TOR, has link to hippo. So how these links are connecting, and how I'm getting this phenotype. So it cannot just be because of this particular thing, but I did not look into it. But yes, for finding out these things, you have to look at the gene, its localization, interacting partners, and go way before you see this phenotype, and find out what are the steps that are going wrong. Look at neurons itself, look at their morphology, maybe there are glia. So this is not just neuron knockdown. This is the whole. This is heat shocks. It could be knocking this gene down in glial cells. So what is the connection? So for any sort, if for any phenotype that you see here, you are just modeling to the end point. But why are you modeling? You're modeling to find out what this gene is doing, right? So that for that you'll apply different tools, like you'll tag it, you will look at all these things that I told you already. So that is what you have to do for each and every gene. So today we just wanted to talk like, how do you model? How do you find out this is the gene which is causing the disease? How do you look at the molecular cellular function? That could be a totally different field, and could be many different classes to find that out.
I see. Makes sense. I think I see what you mean.
But we have good tools here. I mean, as Dipti taught yesterday, she told you about these things yesterday.
Are these one of those mimic lines?
This is, yes, mimic flip-flop. So these are, is it Shiba? Hello? Hello?
Yeah. No, sorry. No,
Okay.
I'm not.
Okay. So this is one of, so I use one of the mimic line and created another fly stock where you could actually flip it to tag the gene, and also flip it back to make a mutant. But yes, it is based on mimic. So it's for conditional gene knockouts in adult flies. Yeah. Falak, I'm going to sit with you for this question because it will take me some time to explain you Kaplan-Meier graph, how to use it, and I'll need a copy pen to explain you. Is that okay? Falak? So she asked me a question and I'll have to sit with her because this is a bioinformatics question and it will take some time to explain properly.
Okay, so she can't talk, but
Okay.
yeah, she is okay. Yeah.
Yeah. Okay. I see. Yeah. Okay.
All right. Thank you so much, Sonal. That was really good. Actually, I also did not know many of these things, so it was very useful for me also.
I hope I was not too fast for students, because I assume that they know many of these things, but we had them in classes. So, but thank you very much. It was really nice. I also learned a lot when I was preparing this talk.
Thank you so much, Sonal, and thank you, everybody.
Thank you, Deepti. Thank you. Bye bye,
Bye.
everyone.
Bye.
Bye.